Good afternoon, everyone. Welcome to today's webinar on nitrosamine impurities, chemistry, detection, and regulatory compliance. My name is Dr. Anna Najafi, and I'll be your moderator for this session. Nitrosamine impurities have been at the forefront of pharmaceutical quality and regulatory discussions since 2018, when NDMA was first detected in Valsartan, and again in 2019 with Ranitidine. Since then, regulators worldwide have issued multiple guidance documents, and manufacturers are now required to conduct thorough risk assessments spanning their supply chain, chemical processes, drug substance, and drug product. In this session, we're going to cover chemistry and structure-activity relationships of nitrosamines, allowable intake limits and CYP-mediated metabolism, detection methods including LC-MS and high-resolution mass spectrometry, practical risk mitigation strategies, and compliance best practices. The webinar is going to run for approximately 50 minutes, and then we're going to wrap up with a 10-minute Q&A session with our technical team. Before we begin, just a couple brief notes. The session is being recorded, so after the webinar, all attendees will receive a link to the recording afterwards. You'll also be invited for a discussion with Dr. Najafi and Dr. Cheu. You can submit your questions at any time using the Q&A button on the bottom of your screen. We'll try to address as many as possible during the Q&A session at the end. It's now my pleasure to introduce our speakers. Dr. Ron Najafi is the founder and CEO of Emery Pharma with over 30 years of experience in drug development, synthetic chemistry, and regulatory science. He's recognized for his groundbreaking research uncovering the root cause of nitrosamine in ranitidine, also known as Zantac, showing it could form NDMA under certain environmental conditions. Under his leadership, Emery Pharma has become a leading FDA-registered, DEA-licensed, CGMP, GLP-compliant CRO specializing in analytical testing, method development, and GMP release testing. Dr. Ryan Cheu is the Director of Chemistry at Emery Pharma. He is an accomplished analytical chemist and pharmacokineticist. He specializes in nitrosamine impurity testing, having developed and validated LC-MS methods for NDMA and related compounds in complex drug matrices that meet stringent ICH Q2R2 guidelines. His work supports GMP release testing, regulatory submissions, and nitrosamine risk assessments for both small molecule APIs and finished drug products. So it's my pleasure to hand it over to our first speaker, Dr. Najafi.
Hi, everyone, and thank you, Anna, for the introduction. I really appreciate you guys are making time to be with us today. I really want to share some of our experience with nitrosamine root cause analysis, testing, and this obviously we've been intimately involved with nitrosamines. But what I'd like to share with you is that nitrosamine, this nitrosamine saga continues. Really started in 2018 with the Valsartan, NDMA showing up in Valsartan, a blood pressure medication that was around for many years without any impurities, and then later a diabetes medication. And then in 2019, we had Ranitidine, Zantac, where NDMA was showing up in large quantities, and this is continuing. And what's important to note is that NDMA is not, or nitrosamines are not new. We had quite a bit of publicity in the late 70s around nitrosamine in food, in cured meat, in bacon, and in fact, sodium nitrite is still present in a lot of cured meat and bacon, which is a potential source for nitrosamines. There are limits for drinking water, nitrosamine presence in drinking water, the cosmetics. Obviously, our focus is drugs. And why drugs are important, because you have patients on drugs for maybe 30, 40 years, sartans or lifestyle medications that people take generally every day. So you have to have controls around nitrosamines. Today, I'm going to talk about possible sources, how to limit them, how they form, mutagenic and carcinogenic potential of these compounds, and also we're going to share with you a little bit of some of what we went through with Valsartan and Ranitidine, and Dr. Cheu is going to talk about how we control, how we analyze, how we can effectively quantitate different nitrosamines. So just as a brief, note, nitrosamines are basically, essentially, it's a combination of an amine. It could be a primary amine, secondary, uh, tertiary, or quaternary. But they all, we end up with is a secondary nitrosamine. And the secondary nitrosamines are generated typically with the presence of an amine and a nitrosonium ion, which is often generated with sodium nitrite and hydrochloric acid. So here is your nitrosonium ion, powerful electrophile, reacting with a secondary amine. So nitrosamines are suspected carcinogens, mutagens. They're class 1 impurity per ICHM-7. And evaluation of nitrosamine in drug products are a big challenge, primarily because of the low levels that we need to quantitate to, parts per billion, nanogram levels, and that's been a challenge for a lot of analytical groups. FDA's expectation from the industry is basically FDA says you're responsible for your manufacturing, you're responsible for your impurity control, you're responsible for quantitating it, having proper quantitation methodology. So it's really the industry, and FDA has provided multiple guidances, the latest of which was in September 2024. There's additional guidances since, and EMA actually just issued a guidance last month. So there are continuously the regulatory bodies around the world, TGAs, coming up with guidances. The traditional assessment for mutagenic impurities have been impurity assessment, hazard assessment, risk characterization, and control. What we need to do with nitrosamines, because they're such a low levels, you have to quantitate down to parts per billion nanogram level, is you have to control where nitrosamine impurities show up. It's really not that complicated. We have to know, okay, sodium nitrites is a big potential source for nitrosation. So we need to control that in fresh solvents. We also need to worry about nitrosamine, NDMA, NDEA, various other small molecule nitrosamines that are very potent carcinogen. We need to look at recycled solvents. We need to look at packaging, bottles, caps, even the ink that goes on a vial, if it's a plastic vial. That ink could harbor some nitrites and then penetrate the bottle and cause problem. Additionally, we need to look at process-related changes. For example, if you're doing synthetic chemistry, and I'm a synthetic chemist, so you have to worry about, okay, it's a multi-step synthesis. You have to worry about all the different reagents, catalysts you're introducing. First and foremost, if you have secondary amine or tertiary amine, you're going to have potential for nitrosamine formation. If you're, intermediates or final products or penultimate steps, you don't have any nitrosamine or you don't have any secondary amine, you're very lucky. But it's really rare to have a pharmaceutical without an amine, a secondary amine or tertiary amine. So you really need to have controls around it, and know the chemistry, and simply Google the chemistry. Hey, am I going to be forming nitrosamine with the AI? You can do a lot these days. And of course, once the drug substance is made and you form a drug product with various excipients, there are excipients that harbor nitrites. So you want to be aware of those. And there are a lot of published data out there, but you have to have some analytical data to show that, okay, your excipient doesn't have sodium nitrite. These are some of the impurity that showed up in sartans, in valsartan, losartans. We were using not we, but general. DMF was being used, triethylamine was being used as it's shown here. Uh, Diisopropyl ethylamine, tetrabutyl ammonium bromide, all of these could potentially get nitrosated and form various very small molecule potent nitrosamines. So these are all the impurities that were found in some of the valsartans that were recalled back in 2018. So basically in 2018, 2019, Valsartan, Losartan came up with a lot of NDMA, and the critical thing is you cannot use HPLC to detect these compounds. You'll be lucky if you could do it by GC. You really need to use GCMS and look for these compounds or do some untargeted analysis by HRMS, high resolution mass spec. Also in September 2019, Zantac began to show nitrosamine, NDMA in this case, and then that's where we got involved with testing. In fact, we were also involved in testing NDMA in Valsartan. And in 2019, ranitidine was showing NDMA by gas chromatography, huge quantities of NDMA. But that was primarily because of the heat that was being generated with GCMS that were forming NDMA. When we switched to LCMS, we were able to see the real quantities of NDMA in ranitidine, Zantac. And of course, simultaneously, FDA was looking at it. Other agencies were looking at it. But I think we were ahead of the game because we were the first one. FDA in December 2019 indicated via a press release that if you have less than 96 nanograms of NDMA in Zantac, you can put your drug back on the market. But we had actually recognized that indeed the Zantac problem was that if you heat the Zantac molecule or if you expose it to moisture, which always gets exposed to moisture and higher temperature, you're going to form NDMA. So it was really part of degradation of the Zantac molecule, the ranitidine molecule. Since 2018, as I mentioned, there have been multiple FDA guidances. Obviously, the September 2024 is most revealing, and I encourage all of you to study that. There have been additional guidances from European Medical Agency, actually last month, and TGA has had some really good ones. There are also efforts in harmonizing that activity vis-a-vis the CPCA, which is carcinogenic potency categorization scheme that all the different agencies are trying to sign up with and agree to, and with industry support, of course, and industry collaboration. Basically, FDA's guidance is look for low molecular weight nitrosamines, such as small molecule NDMA, NDEA various. These are highly potent, highly carcinogenic, and also be cognizant of NDSRI, which is nitrosamine drug substance related impurities. These are nitrosated basically drug substance or basically related impurities of drug substance, it gets nitrosated. So be aware of those and keep in mind that if you have multiple nitrosamines in your drug substance or drug product, the agency requires you to keep the levels really low, below even 26 nanogram per day. Um, so nitrosamines are formed many different ways. The first reaction scheme that I'm showing you is basically showing that chloramines, essentially goes through an oxidation pathway and form nitrosated secondary or tertiary amine. But the most common pathway in pharmaceuticals is you have to worry about presence of sodium nitrite or nitrate, and that could form nitrosamines. The chemistry, if you have primary, secondary, tertiary, quaternary amine, they're all going to be prone to nitrosation. In this case, the primary amine that I'm showing here gets nitrosated and then immediately forms this diazonium cation, this methylating agent, and they gets deactivated. So the focus is really the secondary amine, which it gets nitrosated and it stops at the nitrosated secondary amine. And that nitrosated secondary amine effectively becomes the source of, as it travels through the liver and comes in contact with CYP enzyme, it becomes activated. So why should we worry about tertiary amine? Initially in 2018, there was a lot of speculation. How come we're seeing NDEA, N-nitrosodiethylamine? Uh, where is it coming from? It was coming from triethylamine being present. So triethylamine undergoes a transformation and forms effectively diethylamine or dimethylamine in this case, as it's shown here. So this is a known transformation. I have publication going back to 1978, 1972 for this transformation. This is a very critical slide. It's a little bit busy, but I appreciate your attention. This is really showing the transformation of the NDMA, in this case, to CYP enzyme, with CYP enzyme to the methyl diazonium ion. So this reactive fellow here is very potent. It's a methylating agent. It's like methyl iodide. It's like dimethyl sulfate. It's very potent. And it wants to, it methylates DNA. And it causes basically, it's mutagenic, it's carcinogenic. But what's important is that the CYP NDMA has to fit into the CYP enzyme binding site, and then CYP effectively tries to oxidize and deactivate. So this is CYP's job. The cytochrome P450 and the family of cytochromes, their job is to deactivate any toxins that goes through our body. In the process of deactivating the NDMA, unfortunately, it activates it. So I repeat, in the process of deactivating NDMA, it ends up activating it to a methyl diazonium ion, and that's really the crux of the problem NDMA and other nitrosamines. So NDMA has been the most studied of the nitrosamines. There have been over 20 animal species have been tested with NDMA, and in all 20 animal species, this molecule is tumorigenic. It creates tumor, and and FDA, various agencies, regulatory agencies are extremely sensitive to this because you don't want to expose patients to this molecule unnecessarily. So it's a balancing act between, how much the patients are taking over a period of 60, 70 years, and basically how important the drug is, etc. So in this case, the allowable intake limit, the AI, stands for allowable intake. It's not artificial intelligence, by the way. Allowable intake limits is 96 nanogram per day. NDEA is a little bit lower, 26.5, and so forth and so on. So for those of you who are in generic drug industry, or you're doing analytical, or you're in quality assurance, I would say majority being released uh, using HPLC. And I said that's not sufficient. If we're looking for nitrosamine, here, for example, 40 milligram tablets of nitrosamine, you're looking at basically 96 nanogram. I've done the math here. It comes to about 0.00024%. There's no way we can actually detect that by HPLC. At best, maybe we can detect 0.1%, 0.05%. So we really need sensitive equipment such as LCMS MS, where we do sophisticated, or GCMS or GCMS MS. So those are really the critical instrumentations we ought to have in order to analyze and quantitate these compounds. And Dr. Cheu is going to tell you a little bit more about how we do it and what equipment is used in this case. Basically, all the regulatory agencies are now coming around this categorization mechanism. They call this CPCA, carcinogenic potency, potential for categorization. It's a carcinogenic potency categorization scheme. And the category 1 are the most potent of the nitrosamines, such as NDEA, NDMA. The critical thing for CYP enzyme to be able to actually oxidize these molecules, you have to have an alpha hydrogen. So you have to, in this case, in the case of NDMA, you have 6 alpha hydrogens. And it's also small. It fits into the CYP very nicely, and it gets metabolized to the nasty methylating agent that we talked earlier. There are other compounds. So there basically CPCA does a lot of analysis on, okay, what are the substituents on the nitrosated molecule? For example, in this case, N-nitrosofloxacin. This floxacin is a very well-used drugs in depression and so forth. So you have this related NDSRI, nitrosamine drug-related impurity. Basically, their agency is saying this is pretty potent, 26.5 nanogram per day. And this number could vary depending on toxicology studies that the sponsors or other institutions conduct to see whether indeed this 26.5 needs to stay, or this molecule can be pushed to category 2, for example. Category 2, we have moderate potency. You have 100 nanogram per day. And that's really, again, based on how many substituents. Here we have 5 alpha hydrogen, and then you also have various substituents. And depending on who does the study, toxicology study, the numbers could vary, as is shown here. We're going to share this presentation with you, so hopefully you don't need to take note. Category 3 is 400 nanogram per day, low potency. Again, you have 5 alpha hydrogen next to the nitrogen N-nitroso, but you have substituents that make this molecule safer. So if you have phenyl group, for example, if you have steric hindrance, steric bulkiness in the molecule where it cannot fit into the CYP enzyme, then you have less metabolism, less toxicity. In the case of category 4, you are 1500 nanogram. Here, you don't even have any alpha hydrogen next to the nitroso. So it's, basically this molecule ideally should just pass through the body and not get metabolized. And then, of course, you have this molecule where it's very bulky, very large. You do have some alpha hydrogen, but the regulatory agencies are marking them as very low potency because of their steric bulk. So I want to just jump into a little bit of ranitidine story. You know, this is a molecule that was on the market for 40 years. It was recognized by World Health Organization as one of the safest drugs on the market. But when when we looked at this molecule by GC or GCMS, it started showing a lot of NDMA, and that was really due to the thermal sensitivity of this molecule. And of course, when you look at it by LCMS, you're going to also end up with some amount of NDMA. Uh, European Union, Australia, Korea looked at 150 milligram pills of ranitidine, Zantac, and they found 1,100, 2,100, 8,000 nanogram. Keep in mind the limit, allowable intake limits are 96 nanogram. So this is a huge number. Now imagine you have to take 2 pills a day. That's now you're doubling your exposure. So there have been lots of speculations, and I'm not sure if there's agreement on how the molecule falls apart, but this is one of the pathway where chloramine potentially or oxygen may be involved in nitrosamine formation. NDMA formation. There's also there was evidence of C-nitroso degraded and ranitidine, where they showed that NDMA ranges from 110 to 9,900 microgram per 1,000 milligrams. So we're talking microgram, not nanogram. So and add another 1,000. So there are, you know, this has been accepted by the manufacturer and GlaxoSmithKline and others who tested the molecule in the publication published by Fiona and King that shows that, yes, indeed, relative humidity, temperature, time is responsible for generation of NDMA in ranitidine. And their conclusion was they mentioned that some batches of drugs did not have any NDMA. Uh, they, uh, NDMA was generated in the drug product. Once it was finished, it was formed and put on the stability. The more it stayed on stability study, the more NDMA generated. Exact mechanism is still unknown, and relative humidity, time, and temperature play a big role in formation of NDMA from ranitidine. In the case of Valsartan, blood pressure medication. In fact, my mom was taking Valsartan for many years, and now she's taking something else. But Valsartan was manufactured by Novartis for 30 some years, and they never had any NDMA issues. And we actually tested some of the lots of Valsartan, Diovan, and others, and there were no NDMA. Now, in 2018, lots of NDMA, NDEA, lots of other nitrosamine showed up. The reason for that, which is also publicly available, is that the generic companies started changing the process. During the Valsartan manufacture, you're going from nitrile to a tetrazole, as it's shown here. And this process is best done with a azide. In this case, the original manufacturer used tributyl tin azide, which is a very soft tetrazole forming molecule. But their yield was low. The overall yield of the process was like 50% or so. The generic companies came in to improve that process. During that improving, they went to sodium azide. To neutralize sodium azide, they had to use sodium nitrite. Sodium nitrite it should be a big no-no when you're doing any kind of chemical processing when you got azide. So with sodium nitrite and azide, you form lots of, nitrous acid, and that really begins to nitrate everything, or nitrosate everything, and DMF and everything that was there got nitrosated. So we ended up with a lot of different nitrosamine impurities in Valsartan. So the conclusion was sodium nitrite was a common source. DMF, NMP, all of those that were present. Triethylamine was the precursor for formation of these nitrosamine, and triethylamine indeed reacts with nitrosating agent to form diethylamine and then further nitrosated. So in further in additional summaries, there are basically nitrosamines are observed in drug substance, and basically there are two routes: hydrolytic thermal decomposition of DMF to dimethylamine, nitrosative dealkylation of your secondary tertiary amines, and also nitrosamines impurity typically originates in drug substance, drug product, excipients. So you have to look at all of those to make sure that we're free of potential sources for nitrosamine in your drug substance or drug product. How do we conduct our risk analysis? We look at all the raw materials for the presence or absence of nitrosamines. We conduct untargeted analysis. We need to conduct targeted analysis. We need to look at whether the nitrites, low levels of nitrites are present in the various raw materials, including excipients that are used for the drug product. We look at the chemistry of manufacturing of the product. We look at every step. Do we use secondary amine? Do we use tertiary amine? Do we use quaternary amine? Do we have nitrosating source present? So we carry also a comprehensive untargeted analysis using LCMS, which Dr. Cheu will tell you more about. We also, one of our favorite procedures is this NAP procedure, nitrosating assay procedure. This was actually recommended by WHO, World Health Organization, back in late 1970s, where they recommended that every drug substance should be tested using NAP to look at their nitrosating potential. And of course, if you have volatile nitrosamines, there are a handful of those. I think they should all be looked for and see if there are any of those are present, because they're very potent and category 1 effectively. And ultimately, you have to put quality controls and controls around the various impurities, and consult with toxicologists around categorization of how you're going to categorize your NDSRI or various nitrosamines. With that, I'm going to hand the podium to my colleague, Dr. Ryan Cheu. There we go.
Perfect. Thank you, Ron, and an honor for that introduction. So really, now that we've talked about the background, nitrosamine risk assessments, I want to talk and focus really on some of the unique analytical challenges that nitrosamines bring, how we handle them at Emery Pharma through our process of method development, how we go to method validation, and why that's really required for any sort of regulatory submissions. We're here. Sorry, I'm just gonna hide this. It's right there. Perfect. So yeah, why are nitrosamines such an analytical challenge, right? So as Ron mentioned, it's because they often require very, very low detection limits. So we're talking about limit of quantitations at sub-nanogram level. So this is very low ppb levels, and they're oftentimes in very complex matrices. Really what makes nitrosamines unique is that they actually represent a diverse chemical properties, and they require tailored strategies because of that. So for example, Ron had mentioned things like NDMA, NMBA, MNP, all of which have unique properties, whether they're extremely volatile, very polar, thermally labile, or even hydrophobic in the case of DIPNA. When we talk about those complex matrices, so oftentimes we're trying to do this detection in matrices that contain many excipients, right? Not just drug substances, but drug products. We have degradation products that can occur as a result of instability, as a result of any of the sample prep that we're doing. All of these can potentially cause detection challenges through either coelution, even suppression, or enhancement through our detection capabilities. Because we are focused primarily on these low levels, trace level contamination is a huge concern, right? Because you're focused on sub-nanogram levels, even small amounts of carryover, small amounts of artifact can all contribute to false positives because of that low analytical requirement. And then as we talked about as well, and as Ron kind of touched on, there are stability issues that are of concern. Nitrosamines can form in the presence of heat, light, changes in pH, moisture, all of which can occur throughout the storage process or even throughout the sample preparation process if you're not careful through means such as buffering or quenching. So the big takeaway message from this slide is that detecting them is not just about sensitivity, but it does require selectivity. It does require understanding the stability and very, very careful sample prep. Just want to. How do you move forward? Sorry, there we go. So here at Emery Pharma, we have a diverse range of analytical tools at our disposal. So in this table here, we kind of list out some of those instrumentations, what they're best for, and just some notes that are relevant to that testing for that specific instrument. So the most common is going to be LC-MS, right? And that's going to primarily be best for non-volatiles. So these are polar nitrosamines such as MNP, DIPNA. This is the most widely used instrument. The FDA highly recommends using LC-MS when possible. The one caveat is that it does require clean extractions. So it does require some sample preparation, and you do need to optimize for unique MRM transitions that are going to be specific for your instrument. And we'll get into that in subsequent slides. For GCMS, this is going to be primarily used for our volatile nitrosamines like NDMA and DEA, and that's primarily chosen for its high sensitivity. Something to keep in mind, though, that when using GCMS, you do potentially need to derivatize, and that is a requirement for some matrices. And then finally, as sort of Ron and I think Anna had touched on in the introduction, HRMS is another very powerful mass spec. So this is high resolution mass spec. This really allows any sort of screening, early identification of potential unknowns, and it's really structural confirmation testing. That's going to be particularly relevant for this last technique here, which Ron just touched on in a few slides ago, which is the NAP test. So the nitration assay procedure from the World Health Organization in the late 70s, where you looked at the potential for nitration as a result of your excipients, the excipients themselves, your API. And again, it's not compound specific like some of the earlier techniques, but it is a useful screening early on and as part of that risk assessment. The images below are really just sort of the traditional instruments that we have here, LCMS, GCMS, and HRMS going from left to right. And then finally, that box there in the bottom right is really just the parameters that's done for the nitration assay procedure. So now we want to focus on method development, right? So how does Emery Pharma really handle these unique analytical challenges presented by nitrosamines? So really, we want to break this off into 4 major sections. The first one being sample preparation. So I talked about nitrosamines having unique matrices, oftentimes very complex matrices. So we employ really the traditional sample preparation techniques. Some of the more common ones, bullet point there, LLE, liquid-liquid extraction, SPE for solid phase extraction, or protein precipitation. And again, the analyte is really what's going to dictate what's the best choice for that sample preparation. We use stabilizers as well to help with that instability that we see with nitrosamines, and the use of isotopically labeled internal standards to capture that variability that you may see throughout that preparation process. Once the sample prep is complete, we do inject the sample into our instrumentation where it's subjected to chromatography, right? So that's sort of that second major bullet point right there. where it's important to choose the most appropriate column. And again, that's going to be analyte dependent, where you could be looking at really polar columns for maybe helic or traditional reverse phase C18 columns. We optimize the gradient and flow rate to try to separate out the analyte of interest from any of the excipients or co-eluting compounds that can prevent the specificity that we need for our method development. As we move from the chromatography, we now move to the detection. So in this case, our mass spec. So we optimize our mass spec for a variety of different parameters. Again, the goal being for sensitivity and specificity. So we do that through unique tuning of MRM transitions that are specific to the analyte of interest through product ion scanning. We optimize things like collision energy, gas temperatures, gas flows, sheath gas flows, all of which contribute to the potential sensitivity of the instrument. And we do confirm both the peak shape and retention time as part of that optimization process. And then finally, as I mentioned, one of the unique challenges that nitrosamines bring, because we're talking about very low levels at the LODs, LOQs of ppb levels or sub-nanogram levels, carryover contamination is a huge risk. So we capture that by doing dual needle washes, introducing guard columns, injecting blanks, back flushing, all of which is part of the system maintenance that we do to ensure we are not contaminating or contributing to the false positives that we may see on an analytical testing. So as we move from method development, we want to go to method validation, right? So why do you need method validation? Why is it important? So nitrosamines often sit near that limit of detection level, right? They're very low. So any sort of noise, any matrix interference, any sample prep variation, they can all lead to false negatives, false positives, overestimation, all of which are going to be problematic. Having a validated method ensures that those issues do not arise. So validation is required for CGMP release, ICH stability studies, any sort of regulatory responses. So it's absolutely critical for you to understand what validation is, what it entails. This table below here sort of represents what scenarios where if you don't have validation, what can happen. Again, we talked about false negatives, false positives, where you have unsafe batches being passed, or you have unnecessary recalls. You can have inconsistent results that vary from batch to batch, unexplained deviations, all of which are going to cause you problems. With full validation, you have now a sensitive and specific method for your analyte, oftentimes near that limit of quantitation level, which is going to be highly relevant for that acceptable intake limit that Ron talked about. You have accurate quantitation in real matrices, not just in neat standard. You have repeatable results, and most importantly, you have robust auditable performance that can now be used for submission or responses to any sort of regulatory agency. So this slide here, as Ron mentioned, we will be sharing these slides with all of the attendees. This really captures a lot of the different parameters that we consider when following the ICH Q2R2 for method validation or analytical method validation. The middle column really just represents some of the goals that we have when you do this type of parameter, when you test for those specific parameters. And then again, notes is just sort of a typical range or just typical cases of which we would apply. So for example, for specificity, our goal is to have no interference, right? Whether that's interference from the matrix, interference from degradants. And we do that through testing the unspiked matrix, placebo samples, and having that comparison. Other relevant parameters are things like accuracy, right? So you want to make sure that your quantitation is accurate, typically in that 85, 115%. And you can do this through testing multiple lots to ensure you don't have any of that variability. Things like matrix effects are highly important, right? You want to make sure you understand whether your matrix is causing ion suppression, ion enhancement, and we do that through post-extraction spikes. And then finally, we want to make sure that our instrument is working. So things like system suitability are very relevant in terms of confirming peak shape, making sure your retention time doesn't shift, that it's consistent, and your standard deviation of replicate injections is within reason. And again, the full list of parameters will be shared with you following this webinar. So I do want to walk you through a quick sort of case study to highlight how we sort of do this development, validation. And this is an example of a nitrosamine called MNP, so 1-methyl-4-nitrosopiperazine. So this is a nitrosamine impurity of concern for Rifampin, a common DDI drug that I think both Anna and Ron had talked about. This can form under certain conditions through that piperazine ring. Again, Ron is the synthetic chemist, so he probably can answer more questions about that specific mechanism. But the regulatory agencies do have a limit of about 0.16 ppms, so a little less than about 160 nanograms per gram of MNP because of the carcinogenic potential that it exhibits. What I want to talk about, though, is this actual sample prep and the validation. So for the sample preparation, we actually took Rifampin capsules. We did a methanol extraction, cleaned up the sample through vortexing, centrifugation, filtration, until finally we monitored the relevant quantifier and qualifier transition specific for MNP in Rifampin through our LC-MS. Some of the relevant validation results are highlighted there below, where we exhibited great linearity at greater than 0.999 across a wide range that captures the range of what the acceptable limit intake is. We observed an LOQ of 0.03 nanograms per mil, so this is 0.03 ppb, far lower than what that limit is, giving us the confidence that we can detect low amounts of MNP. What I do want to draw your attention to is actually the in-matrix accuracy component. So what we do here, beyond just a normal accuracy test, is that we spike in known amounts of MNP into Rifampin. So again, we're spiking in known amounts of the analyte we're measuring into the actual matrix of choice to capture and see if we see any recovery issues, any matrix suppression or ion enhancement. And we do this at 3 different levels. Of note for this validation, that spiking at all 3 levels exhibited an accuracy of within 90 to 105%. So again, giving us the confidence that we not only can accurately detect MNP, but quantitate it in the relevant matrix of which you would potentially see it in. So really, it's just sort of on the back end of this webinar, I want to talk about how we partner with our clients on these nitrosamine challenges, and hopefully that's one of you that are attending this conference. But really, there are 4 major components for that partnership. One is the customizable method development, right? So we tailor all of our method development, all of our analytical techniques and instrumentation specific for the APIs, specific for your excipients, your drug products, so you know that our method is going to be developed specifically for you. Following that development, we do also validation per ICH Q2R2, where we talked a lot about those parameters, why that's important, and we went through that case study of MNP. In addition to that, if you're early on and you want to be part of the early stage screening, we offer the NAP test, HRMS, again, things that Ron sort of highlighted as well. And the goal being that we want to provide you with regulatory ready reporting. So that's auditable or audit-ready reports, COAs that can be used for submissions, and really the data just being fully defendable. So in summary, really, we talked about some of the key lessons for nitrosamine analysis, right? So I introduced my portion of this presentation with some of the unique challenges they bring. So again, trace level detection is only meaningful if your method is actually validated in a real matrix or in a real world condition. And that success depends on the appropriate and optimized sample prep, selective chromatography, where you can now separate the analyte of interest from any excipients, degradants, proper mass spec tuning for specificity, sensitivity, reproducibility, and then strict carryover controls, because oftentimes those trace levels, you're at higher risk for contamination and carryover. Again, we highlighted that sort of validated case of MNP. I don't think I need to rehash what those results were, but again, the detection is just the beginning. Really, success means delivering validated data that is fully defendable, that regulators can trust. So yeah, I'd like to thank you all for joining the webinar, for listening and taking time out of your day. I'm going to turn it over to Anna, our moderator, for any questions. I encourage you to reach out. But yeah, I appreciate your time. Thank you very much.
All right. Thank you so much, Dr. Cheu, Dr. Najafi, for the very insightful presentation. I learned a lot. I hope our audience learned a lot today about both the scientific and practical aspects of nitrosamine control. Now we're going to move into the Q&A portion of today's presentation. Okay. Our first question from the audience. In vitro, might you be able to scavenge the NDMA formation by including something simple like histidine or tryptophan in preparation, and perhaps in vivo, use such supplements to reduce NDMA in the patient? So the first question is about scavenging NDMA via, something simple like histidine or tryptophan, and then in vivo using supplements?
Thank you for the question, Patrick. We are obviously, when you get down to nanogram level impurity, it's extremely difficult to scavenge things. If it's volatile, you may be able to take advantage of the volatility of the compound and try to scavenge something, but that's very challenging. In vivo, however, nitrosamine basically goes through your body and goes through the liver, goes through your intestine into the liver, and then gets metabolically activated. So once it gets activated to methyl diazonium, it's a methylating agent. At that point, perhaps some antioxidants might be helpful, but it's very challenging to try to scavenge and or try to effectively fight it in vivo when it's in the body, in my opinion. But I'm sure there are a number of companies who are trying to actually uh, devise various things to pick up NDMA and various volatile impurities, but like I said, it's challenging.
Okay. Please continue to submit questions if you'd like. We have about 7 minutes left for Q&A. The next question from our audience is, what is the best instrument for hydrophobic NNO compounds?
No, that's a good question. Again, it depends on the volatility. You typically will look for GC for more volatile compounds. I'm not sure what specific compound you're referring to that has the NNO. We have access to all of those instruments. We would primarily try, in this case, probably LC base first. But we do have access to a GC if we need to pivot to that approach. Oftentimes, the matrix will also play a role in potentially dictating it. I think regardless of what instrument you choose, the end goal is still the same. You're trying to develop a method that's specific and sensitive for your analyte. But yeah, if you want to share more information about that specific compound, maybe you don't need to maybe do that on the chat in a public forum. Please reach out and we can chat about it and come to an agreement on the best method development strategy and what instrument to use.
All right. The next question is follow-up to our first question. So if you can't scavenge, can you find some additive that drives the reactions in a different direction?
We have to think about it. I think these are very good questions, and please connect with me and directly. All of you can reach out to me on LinkedIn. Please feel free. My email is ron@emerypharma.com. So feel free to reach out. You need to have my email to connect with me on LinkedIn. And also through our website, you can connect with us. We have multiple mechanisms, and also info@emerypharma.com. Love to chat with you. This is an area where we want to put our thinking hat on and see how we could actually prevent formation of nitrosamines or scavenge. And those are all challenging, needs to be tested. We're actually trying a few different things on our own internally as an internal research project to see whether we can scavenge some of the volatile nitrosamines at the moment.
Okay, next question. So if the FDA limit for a drug is to be below 5 ppm, for example, how do you determine the limit of detection or limit of quantitation?
No, that's a good question. So both of those values are primarily dictated by two different sort of branches, I would say. The FDA limit is defined from a safety perspective, right? From the carcinogenic capabilities of the analyte. From the analytical side, determining the LOD or your limit of detection, your limit of quantitation is purely experimental based. So you do the best you can in terms of the sample preparation, the mass spec optimization that we talked about to try to get your instrument to as low as possible. The instruments themselves are theoretically able to get to sub picogram per mil levels. So you should be able to capture whatever LOD or LOQ is typically for those acceptable intake limits that I think Ron sort of captured what those ranges were in some of his earlier slides. But experimentally, we have a lot of different strategies that I kind of touched on to how we can get as low as possible. But that is an experimentally observed number.
Great. Okay. Dr. Cheu, stay right there. I think the next question is for you.
Sure.
So how do you decide which internal standards to use for such low-level quantitation?
Oh, yeah, that's a great question. So I kind of touched on it a little bit, but I can talk more about it. The goal of an internal standard is really to capture variability that you may see in terms of your sample preparation, in terms of the instruments themselves. Because you're dealing with such low levels, having that sort of tracer or that marker that can tell you that variability is absolutely critical. Traditionally, you want to use isotopically labeled standards. So this is your analyte that's now isotopically labeled, whether that's deuterated, C13 labeled. In the absence of those, you typically will try to find a surrogate analyte that mimics the behavior of your analyte of interest, again, with the goal of trying to capture the same chromatography, the same retention time, basically the same thing as what your analyte of interest is, so you can better understand and capture that variability.
Thank you.
Sure.
The next question I think is for Dr. Najafi. You mentioned that nitrosamines can be metabolically activated. Can you comment more on that?
So nitrosamines could be entirely harmless if they're not metabolically activated. So they have to essentially get into the binding site of the CYP enzyme. They have to be small enough and have the available alpha hydrogen and not be sterically bulky. And then they get metabolized into methyl diazonium molecule, which is essentially a very potent methylating agent. So metabolic activation is critical, and that's really where the trend is, how FDA is determining this CPCA, this carcinogenic potential categorization analysis. And that CPCA is derived from some analogous compounds that have gone through toxicology animal studies and have shown tumorigenic potential or lack of tumorigenic potential. And then, of course, you try to see how your compound compares to those compounds. And so I encourage you guys to look at some of the recent guidances from 2024, from September 2024. Also, TGA, Australian regulatory agency, has a very good guidance. There's also CPCA categorization and that harmonization. But best of all is contact us. We can brainstorm together, help you with risk assessment for your drug product, drug substance. The worst thing is to spend, we just had a recent conversation with a client where they had manufactured 3 lots of their drug substance, each one worth $1 million plus, and their drug is contaminated. And it's just so we need to do those things well in advance to prevent these $3, $4, $5 million loss, not to mention the lost time.
All right. Well, it looks like that's all the time we have today. Thank you so much for attending today's webinar. We'll be in touch with the materials, and please feel free to reach out if you have additional questions or if we didn't get to your question. Thank you so much.
Thank you. Thank you.